You've probably heard the saying, you are what you eat. But have you ever considered if that simple phrase holds the key to your mental health and asked yourself, does depression, particularly when experienced during old age, actually start in the fridge? Today, we're going to explore this together by diving into a fascinating 2022 paper by Dupree's and colleagues and what they discovered about the connection between our food, our blood, and our brains. Stay with me, because once you see how deeply your plate and your mind might be linked, you might not look at breakfast the same way again. Okay, let's take a look at the problem. We're talking about major depression, specifically late life depression in older adults. It's a huge and complex human puzzle that scientists are trying to solve. Dupree's and her colleagues focused on this group of people because late life depression is a major public health concern. It's not just about mood. In reality, it's strongly linked to memory problems and a higher risk of dementia. The trouble is, our current medications are struggling to keep pace and they don't work for everyone. This is why we urgently need new options. And that's where lifestyle factors like our diet come in. This leads us to our big question. We know diet might be linked to late life depression, but how? How are our diets connected to our deepest feelings? The story starts inside the brain. This study looks at a process called neurogenesis. That's the birth of new brain cells in the brain's memory hub, known as the hippocampus. The core idea is clever. This cell nursery is right next to your blood vessels. It's constantly listening to all that is circulating in your blood. And since your diet influences what's in your blood, the researchers asked, can the blood from older adults directly influence this cell building process? How did they test this? Since we can't exactly peek inside a living human brain to watch new cells being born, the researchers used a clever method. They took blood samples from 373 older adults. Then, in a lab, they fed a tiny drop of the liquid from that blood, called the serum, to human brain stem cells growing in a dish. This allowed them to watch and see, how did each person's unique blood environment affect how the new brain cells grew? But they didn't just watch. Here's what they did with the data's insights. Dupree's and colleagues first used statistics to see how changes in the birth of new brain cells, or neurogenesis, were connected to both late-life depression and the specific nutrients and molecules found in the blood. They used methods like logistic and linear regression to measure how strong these connections were, and other, more complex methods like mediation and moderation analyses to figure out how or when one factor might influence another. These methods were essential for checking and adjusting for other known issues, which the researchers kept a close eye on. These potential issues or confounders are age-related brain changes, inflammation, stress, or a drop in thinking abilities. They also accounted for things like age, gender, education, and starting health differences, and used special corrections to avoid false positives and make their findings more reliable. Finally, they compared these results from the dish against the participants' mental health history over a long 12-year period to see who had no depression, a single episode, or long-term late-life depression. So what did they find? The first key finding was the way late-life depression showed up varied from person to person. Two main groups were identified from the research. The first group of people experienced one depressive episode during the entire study, whereas the second group of people experienced constant or recurrent depressive symptoms. See this sad face drawn here in the sand? Yes, it's real, it's painful, but it's temporary and on the surface. The researchers first noticed they had reduced brain cell death, but then they figured out that this finding was actually being confused by the overall size of the hippocampus, which is your brain's memory hub. This means that once they took the size of that memory center into account, the specific changes they saw in the brain cells for this group weren't really the main independent cause of late-life depression after all. What it boils down to is that for people with a single depressive episode, the root cause is more likely something external, like life stressors related to things such as education. This is not the same as chronic late-life depression, which we will touch on next. Now, recurrent late life depression is not just a sad face in the sand. It is something much deeper, persistent, and more profound. For individuals with chronic late life depression, the researchers found that their blood told a different story. They discovered an issue in their blood that affects their brain, which negatively impacted the way new brain cells grow. So instead of healthy cells, this increased growth resulted in new brain cells that were badly built, meaning they had shorter, fewer branches and couldn't communicate well. Most importantly, this damaged cell growth was linked to those who also had memory or thinking problems, which is known as cognitive decline. This was demonstrated as the biological marker, named MAP2, for this faulty cell growth was only high in individuals with late-life depression and symptoms of cognitive decline. This suggests that underlying issues in the body's functions, like metabolic factors, might be interfering with their ability to think about and remember things. You might be thinking, why does this matter? See, for a long time, we might have been told that depression is just a chemical imbalance in the brain. However, this study suggests something new, especially on late-life depression. Imagine your brain as a construction site, always building new houses and fixing old ones. Now, for some people with chronic late-life depression, the builders in the brain don't work as well and move in slow motion. And in this study, the researchers found that this building problem might be affected by what's in your blood, which is influenced by what you eat. Specifically, they found two important things in the blood. First is a type of fat called PC35. Second is a tiny energy-related molecule called butyryl carnitine. Now here's the cool part. The construction site in your brain has a job called neuronal differentiation, which means it turns young brain cells into useful grown-up brain cells. And the scientists discovered that how well your brain does this cell-growing job affects the link between the PC35 fat and how depressed someone feels. And the link between butyryl carnitine and late-life depression also changes depending on how well your brain does this job. So the crucial takeaway here is that your diet might not be as passive as you think. Because the factors in your blood from the food you eat are likely forming a connection between your plate, your blood, and your brain. In other words, it is possible that what you eat goes into your blood and affects how your brain builds itself, which affects how you feel. So, what can we do? One thing we can do is to prioritize having a well-balanced, healthy diet, consisting of foods rich in fiber, such as whole grains, beans, and vegetables. Here's why. Remember one of the important molecules we spoke about, butyryl carnitine. It actually comes from butyric acid, which is made in your gut when good bacteria digest fiber. This means when you eat foods rich in fiber, your gut might help your brain's construction site by giving it better, sharper tools to work and build with. Other research has also shown that the Mediterranean diet, which includes fresh and whole foods and limits red meat and processed foods, might reduce the risk of chronic late-life depression. Of course, no single study is the final answer. We must acknowledge the limitations. First, we have to be cautious in interpreting results and remember that what happens in a dish might not perfectly reflect what happens in a complex live human brain. Second, this study shows a powerful link, but because they only took a snapshot of brain cell growth at one moment, we can't be sure that one thing causes the other. It's a bit of a chicken and egg situation. We don't know for sure if the faulty cells cause late-life depression or if depression causes the faulty cells. Finally, future research needs to expand this model to include other important factors, like the brain's immune cells. So to wrap up, Dupree's and colleagues have revealed that depression, specifically late-life depression, might not only be due to a chemical imbalance in the brain. In fact, it might be connected to our entire body and the food we put on our plate. The study teaches us that factors from our diet circulating in our blood are likely affecting the basic process of building new brain cells. And what this means for us is that diet changes might be a practical way to influence how new brain cells are made and potentially lower the risk of depression in later life, which ultimately suggests that the simple everyday act of eating might be one of our most powerful tools against depression in later life.